My name is Dr. Sumangla. I am here on behalf of Sri Chaitanya Saraswati Institute and Princeton Bhakti Vedanta Institute. Today, I am honored to have the privilege to introduce our first speaker, Professor Denis Novel. Professor Emeritus and co-director of Computational Physiology at Oxford University. One of the pioneers of system biology and developed the first viable mathematical model of the working heart in 1960. Over 350 articles, he published over 350 articles in academic journals, including Nature, Science, and Proceeding of National Academy of Sciences, PNAS, and Journal of Physiology. Today, he is going to talk and his topic of his talk is purposive explanation are more useful for identifying lower level activity in living system than the other way round. I request to Professor Denis Novel, please, you can start your presentation.
Thank you. Shall I now share screen?
Yes, are you able to?
Go ahead and see whether that will work. You need to give me permission, I think.
Yes.
Yes, okay.
You should be able to share now.
Right, I'll try again then. Right, here we go. And what I need to do is to make sure we're getting that right. You can see my screen with its, and we'll start playing.
We're not able to see anything yet, Professor Noble.
Okay, I'll try again. There's something clearly not. I must have neglected to press a particular button. I'll just try and see what that is. Let's go to share screen. Share. There we go. Does that work?
It does, yes.
Good, excellent. Right, we now go to full screen, sharing the slides. That's hopefully now clear.
It is, yes, it's full screen.
Okay, I'll go straight ahead then.
Let me see, but I do not. You see,
all
right. Continue. I'll try to figure it out.
Okay, it seems that it's working for some people and not for everybody. We're a minute or two before I actually am due to start, so why don't we just make sure that these work?
Yes.
Okay.
Dr. Huera, are you able to see the shared screen?
Yeah, I can see you. That's Kishore.
Yes, we can see.
Okay, very good. I'll proceed then. I believe that the lecture I'm going to give you this morning is absolutely relevant to the interests that you explained at the introduction to the meeting, because I'm going to give you the best test that any scientist can as to whether or not purposive explanations are effective. That is, are they good at predicting other levels of organization, and how good are they compared to the reverse process of trying to predict high levels of organization from lower levels of organization? And in fact, what I'll do there is to refer at the end of the lecture to the latest study in GWAS explanations of health and disease, showing that they're almost useless. By contrast, purposive explanations have been shown by physiologists for many years, nearly 400 years now, to be very effective at explaining lower level living processes. And that will consist in me explaining how William Harvey, who worked in the 17th century on the circulation of the blood, and demonstrated the circulation of the blood, also made a major prediction that was confirmed by microscopy at a later date. I will also give examples from the kidney system and what is called countercurrent flow. And then from the 19th century, Charles Darwin's work on evolution and the theory of pangenesis, which included his idea about tiny particles. We'll come to that at some point. And finally, Hodgkin cycles and downward causation. The function of the circulation was the object of William Harvey's famous book, De Motu Cordis, on the movement of the heart and blood in animals. He published it in 1628. It was the first use of quantitative calculations to answer a functional question, and is therefore one of the origins of the quantitative systems biological approach. And his discovery of the function of the heart as the circulation of the blood was already announced in a lecture he gave about 10 years earlier, when he wrote, "So it is proved that a continual movement of the blood in a circle is caused by the beat of the heart. And knowing how frequently the heart beats and what volume of blood could be pumped out during each beat, he estimated how rapidly the heart could empty the entire volume of blood in the body." He wrote, "This is also clearly to be seen by any who watch the dissection of living creatures. For not only if the great artery be cut, but as Galen proves, even in man himself, if any artery, the smallest, be cut in the space of about half an hour, just 30 minutes, the whole mass of blood will be drained out of the body. By careful reckoning, therefore, the quantity of blood forced up beyond the valve by a single compression may be estimated, and this multiplied by 1,000 gives so much blood transmitted in this way through a single portion of the veins in a relatively short time that without doubt you will be easily convinced of the quickness of its passage of the circulation of the blood." And the answer in the case of a human is indeed around 30 minutes. William Harvey then proposed an answer to the question, where does all this blood in the body go? Because until Harvey, people thought that the blood ebbs and flows back and forth as it gets pumped into the expanding arteries, rather like the tides of the sea, ebbing back and forth on the seashore. Harvey realized this could not be true, since he demonstrated one-way flow of blood through the veins, taking blood to the heart, because the veins have valves, so ensuring that the blood cannot travel back to the extremities. It must always flow through the veins towards the heart. And he concluded, it must therefore be concluded that the blood in the animal body moves around in a circle continuously, and that the action or function of the heart is to accomplish this by pumping. This is the only reason for the motion and beat of the heart. But then he did another prediction, because he saw that since there is continuous one-way flow, the arteries and the veins must somehow connect together. Now, with the naked eye, Harvey could not see how that happened. So he supposed there must be what he called porosities, little holes in the tissues through which the blood could flow from the tips of the arterial tree to the tips of the venous tree. Harvey's achievement, therefore, was not only to discover the circulation of the blood, but also to make a prediction that he himself could not prove. And it was around 50 years later, in 1661, that Marcello Malpighi used a light microscope to observe the flow of blood through what we now call the capillaries in the brain and in other organs of the body. So, however much we might study the particles of blood, what we now call cells, platelets, vesicles, and all their associated fluids and dissolved molecules, nothing in these molecular movements could tell you that blood circulates. And this was realized by Benedict Spinoza, the philosopher, who communicated his idea to in a letter to the Royal Society in 1665. And he wrote, let us imagine, it was done in Latin, as a matter of fact, concipiamus si placet. Let us imagine, with your permission, a little worm living in the blood, able to distinguish by sight the particles of blood and lymph, and to reflect on the manner in which each particle, on meeting with another particle, either is repulsed or communicates a portion of its own motion. This little worm would live in the blood in the same as we live in the part of the universe, and would consider each particle of blood not as a part, but as a whole. He would be unable to determine how all the parts are modified by the general nature of blood and compelled by it to adapt themselves so to stand in a fixed relation to one another. Now, there is an important lesson here, which we'll see repeated as I take the other examples of purposiveness in physiology. Functional theories about the workings of the cells, tissues, and organs of living systems can make micro-level predictions that can be tested experimentally. But as Spinoza realized, the converse does not necessarily or even usually hold, because the multitudinous directions of movement of particles will not yield conclusions about the functioning of the whole. As we well know from thermodynamics, where the constraint of such movements by containers generates the high-level properties of temperature, volume, and pressure, and where most of the lower-level stochasticity is ironed out. Well, biology has reached a similar impasse in seeking to explain physiological function in health and disease purely from measurements of gene sequences and associations in genome-wide association studies. I will refer to that failure at the end of the lecture. So, the constraint of molecular and other microcomponents by membranes and other boundaries in physiological systems can be readily understood with a simple diagram, which I'll show in the next slide. Without constraint, particles will move every which way, and so disperse any functional networks that might perform. But under constraint by boundaries, functional networks may persist. And I think that such constraint will have developed at the very early stage in the development of life. Let's go straight on to the diagram. It's a very simple diagram from a book that I will refer to at the end of the lecture, Understanding Living Systems, just recently published by Cambridge University Press. And the point is very simple. In the little diagram on the left, all the particles can be moving every which way. And some will be going up, some to the left, some to the right, some down. And what will happen, of course, is that any network that formed of interactions between those particles will simply disperse. But as soon as you constrain those particles by a membrane that only lets particles to go through when there is a need for them to do so, you can then keep a functional network together. A functional network may then survive. Constraint, where does that come from? It comes from higher levels of organization. The constraint in cells, tissues, organs of the body are always purposive. They have a function. Now come to the other example, a much more recent one, which is the renal system, the kidney system, and what is called countercurrent flow. because physiological studies of the kidney and the renal system illustrate the same kind of functional analysis, leading to successful predictions of micro-level components and processes. Because, as you can see in my diagram, the structure of the kidney is rather like a compact maze, with tiny tubules interweaving with each other, forming pathways bending back on themselves to run from where they came from. Now, from a molecular viewpoint, this all seems pointless. Why carry fluids and molecules dissolved in them back and forth through such long tortuosities? Well, the answer is that calculations using physiological high-level insight provide the answer. From an engineering point of view, the arrangement of the renal tubules uses the principle of countercurrent flow, which was first proposed actually in the kidney by Werner Kuhn in 1951 and confirmed by Gottschalk in 1998. And engineers frequently use this principle to construct heat exchangers and to concentrate chemical compounds. Now, this principle is of functional use all over the body. It's amazingly simple, but also deeply explanatory. In the limb extremities, arteries and veins run together side by side. So the hot blood emerging from the heart warms the cool blood coming back through the veins. This heat exchange system conserves heat in the body as a whole, so enabling warm-blooded organisms to keep heat balance more easily. And this balance, in turn, optimizes metabolic processes such as enzyme reactions, so that they run at optimal levels. And furthermore, these functional insights predict the existence of the required selective protein transporters in the cells lining the kidney tubules, and many of those have now been found. Now, I want to make an important point here. Those predictions will not tell you which particular kind of molecule will perform the function. They will identify for you the functions that are necessarily served by the molecular level. You can then use that to look for precisely those kinds of functions in channels in the body. Now I come to the third example, Charles Darwin and his theory of pangenesis. Because in Charles Darwin's 1868 book, The Variation of Animals and Plants Under Domestication, he outlined a theory for how physiological changes during their lifetime could influence future generations through their effects on the germline cells, the future eggs and sperm. And so convinced was he that he treated his theory of pangenesis as what his biographers later called a beloved child, because this was no passing fancy. He very much wished it to be true. And at the same time, he realized its speculative nature, since no one had any evidence that transmission of material, which he called gemmules, from the body, the soma, to the germline, as the future eggs and sperm, could occur. And he wrote, physiologists maintain, as we have seen, that each cell, to a large extent dependent on others, is to a certain extent independent or autonomous. I go one step further and assume that each cell casts off a free gemmule, that's the word he used for these particles, which is capable of reproducing a similar cell. And he clarified the existence of free gemmules is a gratuitous assumption, yet it can hardly be seen as very improbable, seeing that cells have the power of multiplication through the self-division of their contents. He therefore imagined his idea of gemmules as rather like spores. He was wrong to suppose they were capable of reproducing a similar cell, but correct to see the cells as casting off a free gemmule. His text only needs revising to read capable of influencing other cells, for his gemmules to become, well, the extracellular vesicles which we found today. We'll come on to those in a moment. His theory did not need them to reproduce, only to influence characteristics. And here is the confirmation. There are many confirmations now published in the recent literature showing that particles do emerge from soma cells all over the body, as you can see in this diagram, moving down towards the future eggs and sperm in the germline. Now, this is just a schematic diagram, but what the article shows, and I'll quote the article at the end so that you can follow it up if you wish, is that the emerging role of extracellular vesicles, which is the word we would use for gemmules today, is that they contain RNAs in short and long-range soma to germline communication, so that lifetime factors, including nutrition, stress, and disease, induce the production and release of these extracellular vesicles containing various RNAs into the circulatory system, including from the brain after surpassing the blood-brain barrier, and then the tissue EVs migrate through the blood follicle barrier and the blood testis barrier and enter the antral follicle. So this diagram is from an article published with Daniel Phillips, my co-author, just this year, the Journal of Physiology. Hello? Is somebody trying to contact me? Okay, I'll carry on. So when, after Darwin's death, Weizmann proclaimed the all-sufficiency, the Allmacht in German, of natural selection and the necessity for a barrier, it was difficult, if not impossible, for defenders of Darwin's idea to resist Weizmann's exclusion of the inheritance of acquired characteristics. And yet, in retrospect, we can now see that Darwin was using a similar logic to that of William Harvey and his porosities. The fact that we can't directly see something postulated by a valid scientific theory does not in itself disprove the theory. It may simply mean that the resolution with which we can see very small things is not sufficiently high. Harvey's porosities appeared just 3 decades later in the light microscope detection of capillaries. Darwin's gemmules have had to wait much more than a century, but we have nevertheless found them. And the evidence that DNAs, RNAs, and other molecules get transmitted from the body to the germline is now clear from work in very many laboratories. Now, finally, as my examples from physiology, I come to the Hodgkin cycles and downward causation. Because Alan Hodgkin, who was very famous in 1952 for working with Andrew Huxley to produce the first model of nerve activity, the nerve impulse, he did so by relating the action of protein channels to cell electrical potential. And it was this cycle of the cell potential influencing protein channels, which then create current, which influences the cell potential, and so around in a circle all the time. That was published in 1952, and it got Hodgkin and Huxley the Nobel Prize for Physiology and Medicine in 1963. But it also led to me, as a young student at University College London, creating this model of not nerve, but of the heart, and the very much slower impulses that develop through, first of all, entry of sodium through a channel that leads to depolarization of the cell, which leads to the spike-like response, and then slowly, as a consequence of exit through a potassium channel, makes the cell become negative again. And excuse me. And so the process continues as a cyclic event indefinitely. It never stops. Now, what that shows is, once again, a high-level property, which in this case is a property of the complete cell. The electrical potential is generated only by a complete cell. Without the cell membrane, there would be no potential, is absolutely necessary to enable you to explain events at the molecular level. Now I come to a use of this approach to, as I put the title here of another lecture I gave a year or two ago, also in the United States, I called it Pacing the Heart to pace the pace of evolution, because heart rhythm is a very good example of a difference that's going to become very important towards the end of my lecture, which is the difference between association of genes with function and causation. They are not the same. And most GWAS studies, that's genome-wide association studies, make the assumption that they are the same. We'll see that in a moment. Now, the example I'm going to give is one that identified an HCN gene, don't worry about the technical detail, which contributes, as we found, about 80% of the electric current that generates the rhythm. Whereas knockout of the gene or block of the protein that it enables to be produced changes the frequency of beating by only 10 to 15%. And here is the proof of that claim. What I'm showing here is the HCN channel, which is called IF, carrying a fairly small amount of current in this case, with a large amount of current carried by a background sodium channel. And what you'll notice is as we knock out the large current flow channel, what is happening is very small change in frequency, because what happens is that the HCN channel shown in black here is taking over from the sodium background channel. What that means, therefore, is there's a huge difference between causality. What is obvious here is the HCN channel eventually becomes the greatest contributor to the function, that is, the pacemaker rhythm of the heart. But there's a huge difference between that causal role, which can be as much as 80%, and the association level, which may be only 10 to 15%. Incidentally, we can also show physiologically why that switch happens. It's entirely because of the small hyperpolarization, that's increase in negative membrane potential. I don't have time to go through the details of that, but it does fully explain why the changes occur in the way they do. Causal physiological analysis, which is necessarily functional, that's what we mean by being causal, also enables you to give explanations right down to the molecular level. Now, how extensive is this process of a difference between cause and association? This beautiful study, done just over 15 years ago by Hillenmeyer and his colleagues, looked at all 6,000 genes in the tiny organism yeast, uncovering, as they say, a phenotype for all the genes. And they found that 80% of the knockouts in normal physiological medium, that is, where the yeast can reproduce effectively and live very easily, 80% of the knockouts are silent. But that does not mean they weren't functioning. On the contrary, they showed by taking out components in the physiological fluid necessary for the yeast to function, what the role of those components would be in circumstances in which the yeast is deprived of those components. It gives robustness to the yeast metabolism and enables it to survive in even unfavorable circumstances. Now, this is more or less universal. This paper, published in 2017, just 5-6 years ago, well, it's called the omnigenic hypothesis. It's already had over 1,000 citations, and it shows very clearly, first of all, that all genes contribute to many or even all functions. Many genes also, we'll come back to this point at the end of my lecture, show very low association levels, almost useless for predictive purposes. Third, that the linear addition of such data is not valid, and therefore gene-centric views of development and evolution need completely revising. They wrote, we propose that gene regulatory networks are sufficiently interconnected that all genes expressed in disease-relevant cells are liable to affect the functions of core disease-related genes, and that most heritability can be explained by effects on genes outside core pathways. That means you cannot do an analysis from just a few genes to understanding the state of health and disease. Now, I finish by coming to the reverse of what I've described so far. Let me just summarize what I've described so far. I've given you 4 or 5 examples in detail of how high-level physiological function, identifying, for example, the role of the heart in the circulation of the blood, the role of the kidney tubules in creating countercurrent flow, the role of Darwin's gemmules, which we've now found in ensuring continuity of communication of characteristics in the organism, at least partly to the inheritance of the later generations, and with what are called Hodgkin cycles, that is the electrical activity of cells in the organism, all of which depend on high-level cellular properties at the least, and even even bigger than that. In the case of the heart, of course, it's the whole heart that has the function of showing the properties that lead one to say that the heart's function, its purpose, is to circulate the blood. Now, I referred earlier on to the omnigenic hypothesis, which is the theory that most genes contribute to nearly all functions, and therefore we already know, and for about 10 years have known, that the association scores in genomics are so small as to be usually pretty useless. So what have people done? Well, they've created what I said was not correct. They've created polygenic risk scores. The idea is very simple. The score of association between the presence and absence of a particular gene is so small that it would be useless as a predictive property, maybe just 2 or 3% above the standard rate in the population as a whole. This study takes those polygenic risk scores, that is, adding them all together, to see whether even so, even if it's not, it can't be said to be accurate, because polygenic risk scores add all the risks together. And as I said earlier on, you can't really do that. It's not really valid. But even if you do you then get practically a useful prediction of who is likely to get heart disease, who is likely to have vascular disease, who is likely to have cancer, who is likely to get Alzheimer's? Well, the results are exceedingly disappointing. This is a paper from University College London, published just recently in the British Medical Journal Medicine. And they write, all I need to do is to quote their conclusion. our results showed the poor performance of polygenic risk scores in population screening, individual disease prediction, and population risk stratification. This finding is not obvious from the metrics reported in the polygenic score catalogue. That's absolutely true, but it's clear based on the appropriate metrics used in this study. Our conclusion is consistent with that of others, like the fact that the polygenic theory leads to it being very difficult to predict from particular genes to what we can call function. So their conclusion is consistent with that of others, but is insufficiently recognized. Well, it needs to be recognized because this test uses the most, how best to put this, the most useful test we can use for any prediction from a molecular to a whole organ or whole organism function, which is the clinical trial. Because what they did in this study of polygenic risk scores is a kind of clinical trial. They used the information from the risk scores as though these were the scores from a drug being used to determine the causation of by a particular drug. And what they're showing is, of course, it's useless. You get, for each of those major diseases that I outlined, vascular disease, heart disease, cancer, neural disease, you get almost as many false predictions as you get correct predictions, and that is almost useless. Now, I finish this lecture with a short diversion. Established, therefore, because I think the lecture shows this very clearly, that purposes come from high levels of the organism, influencing the constraints on the lower levels. This also is a diagram from the book that's just come out in Cambridge University Press, Understanding Living Systems, going all the way from the sociotype, that means the social relations at the very highest level of social interactions, the ecotype, that is the niche in which we live, the phenotype, which is what we display as actual functions of the organism, the organ types, the tissue types, the cell types, the karyotype, and then finally, at the bottom, the DNA. And what this diagram shows is that actually there are increasing constraints by all those higher levels on the most molecular level. That is why DNA sequencing will not give you good prediction of the higher levels. Those sequences are constrained by the higher levels. They can't possibly, therefore, be predictive of what their constraints are doing. And that, in a nutshell, is why the Hingorani and others publication in the British Medical Journal is so clear about the outcome. You cannot go that way. It does not work. And yet we've spent billions trying to prove that it does work. We need to move away from that false and already demonstrated false assumption in biology. So where do the purposes come from? They can come from any of those higher levels, because they can arise as forms of causation at all levels of the organism. Note, though, with the sole exception of the molecular level. There is no real causation at the molecular level. The molecules, such as DNA, can't have functions because they're chemicals. They must do simply what their chemistry makes them do. Whereas, just to illustrate that with the case of DNA, DNA is constrained by all the living processes that control it. It is therefore the most constrained level of organization. The really open level is, of course, the sociotype. That is where our freedom to act, our decisions in relation to others in our societies comes from, of course. So I've referred to this little book, which, excuse me, published just 2 or 3 months ago. And we finished the book with a plea for the scientific community and to society in general. And that plea is to the next generations, because it will require creative ingenuity. After 50 years of being taught the wrong way round, to shift the culture of biology away from the misunderstandings of the 20th century. And it will be for a new generation to discover and create their own culture fit for the challenges of the 21st century. And they will have plenty of looming signposts to warn them what went wrong, because theirs will be the generation that must take responsibility for the way in which the Earth's ecosystems need rescuing, even for our own species to survive. That's one of the reasons why this is urgent. And theirs will be the generation that faces the challenge of aging societies, requiring medical science to find solutions to the diseases of old age that do not readily yield to reductionist gene-centric solutions, since those diseases are multifactorial. Only an integrative approach that understands those multifactorial interaction can possibly hope to address those diseases. And that's the second reason why this is urgent, because the health costs of the diseases of old age are now ballooning into levels that could destroy our health systems. I'm not exaggerating. Theirs will also be a generation that can try to recover from the damage to society, the results from reductionist models of physiology and evolution that have metaphorically shaped ideas and models in fields as diverse as economics, sociology, philosophy, ethics, politics. The list goes on, because no aspect of today's society can have escaped dogmas like we are born selfish, genes created us body and mind, it's in his DNA, and the myriad of other tropes of related types that we now use almost without thinking. Fourth urgent thing, those future generations will also need to rewrite the textbooks, not only because they see the virtue of, to quote Richard Dawkins, let us therefore teach our children. That's his way of getting out of the dilemma of the selfish gene. If all we do is selfish because of our genes, how on earth do we counter that? He says we have to teach our children. I don't know how he knows how we can do that. But anyway, but also because their politicians, economists, sociologists, and philosophers will also need to find new strategies in collaboration with biologists who can lead them out of the gene-centric impasse. It is arguably a challenge the scale of which human society has never faced before. And we finished that book with a wish. We wish them all well, and it's a heartfelt wish. Here's my promised set of articles. I hope the organizers of this meeting can feel free to circulate my slides to everybody who is registered for the meeting. And in fact, there's nothing confidential about anything in my talk. The first article is an article showing that physiological selection of environmentally acquired traits can cause speciation. That is purposive. Evolution is not supposed to be purposive. Those of you who've learned it at school will know that. Well, it's not true. Physiology will actually lead speciation. The article showing how physiology vindicates Darwin's dream, that is his pangenesis dream, was published just a year ago in Experimental Physiology, and the full details on how Darwin's gemmules, that is the extracellular vesicles, bubble their way beyond the barrier, has just appeared in the Journal of Physiology. This article, which appears in a Russian journal, translated as Integrative Physiology, shows how purposive views of living systems, physiological views, naturally must now return to the center of biology. And finally, the little book that I published with my zoologist and philosopher brother, Raymond Noble, Understanding Living Systems, was just published around 3 months ago by Cambridge University Press. And everything in that book is explained in lay language. There is no technical detail in that book except the very minimum to describe what a genome really is. Thank you very much. That's the end of my talk, and I think I can, at this point, stop the share. Thank you very much indeed.
Thank you very much, Dr. Noble, for your detailed, in-depth, and inspiring conclusion for the next generation to take seriously. And there's a wealth that you've presented that we can't comment on immediately, but we hope that some valuable questions may come your way during the next 20 minutes, if that's okay with you.
That's okay, yes. I'm happy to take any questions that people might send, particularly via the meeting chat line. And I noticed, so a link to the slides is already up there on the chat line.
Yes, we have just shared your slides on the chat, accessible to everyone here in the meeting.
Yes.
Srila Bhakti Nishkam Shanta Maharaj, Dhanavat Pranam.
So now, can we ask one question?
Sure. Certainly, yes. Please go ahead.
Krishna Keshava Prabhu, what
Yes.
is the schedule? Now we ask the question or later?
Yes, so for the next 20 minutes, we have open Q&A.
Okay, okay. Thank you, Dr. Denis Noble, for your nice presentation. Appreciate it very much. So I would like to ask you to clarify, as many people get the impression in common people that DNA has a very special power inside the cell, and as if it is mechanically controlling a cell by what you can say, its representation as a functional or informatic molecule. So I always try to give example when I present to the students from what we learned from our teachers, that hand does not do anything. It is the person who is using the hand to many things, writing or shaking the hand and things like that. So what we have learned from the advanced cellular biology, that the cell is doing the things which looks as if magical. The molecules are talking inside the cell, but the molecules that have inside the cell by themselves don't have that capacity to do by themselves. Those are miracles. So we want to know your view on that, and also to get some clarification. Like we have a robot, there are some program. Do we still believe that there are some hidden codes and programs which actually controls various functions of the organism, like biologist gene or a religionist gene and things like that?
I like all of those questions. They are the ones we take up directly in the book, Understanding Living Systems. The answers are very simple. DNA was described as the book of life. DNA was described as the program of life. DNA has been described as what creates us, body and mind. That is Dawkins' view. I debated with Richard Dawkins a year ago. He couldn't deal with any of the points that I made, showing that molecular biology itself, not high-level biology like mine, but molecular biology itself has deconstructed those false conclusions. There is no book of life in the DNA. There is no program of life in the DNA. I'm a computer programmer. That's how I produced the rhythm mechanism of the heart as a calculation back in 1960. I used a very primitive valve-based computer, one of the earliest computers available for people to use. I used that in order to show that it was possible for the cell voltage to interact with the membrane channels to produce the rhythm. And I did that by programming. Now, what you have in programs is choices, because you have to allow for different possibilities. If this happens, then that. If not, then something else. You need what I call those if, then, else clauses. You will look through the sequence of C's, G's, A's, and T's until you're completely silly looking through the C, G's, A's, and T's. You will never find a sequence that is the equivalent of if, then, else. There are no choices in the genome. There are switches. Each gene coding part of the DNA has a structure close to it, which, if activated by the rest of the cell, notice that it's the living cell that does it. If activated by the rest of the cell, enables that gene to start informing the ribosomes in the cell to produce the protein necessary for that function. But that's not something that the gene does without being activated from the high-level property of the cell as a whole. So the functionality comes at a different level. It's not at the level of the nucleus. The nucleus is just a membrane that surrounds the DNA and holds the DNA tightly bound together in its chromosomes, usually, and just liberates little bits of it when you need to make more protein. That's all it is. It's the end of story. It is not what creates life. Now, I think for roughly 20 years since the sequencing of the genome, the first sequencing of the human genome in 2001, we've all labored under the illusion, because it is an illusion, that that promise will one day be satisfied, that people will find that program in the DNA and show how it works to create the organism. What they've been presented with from that study at University College London that I referred to, just recently published in the British Medical Journal, is that it's useless, even at predicting which diseases we're going to have. You can go to 23andMe and many other companies and pay maybe $1,000 and get your genome fully sequenced. It might tell you you've got a distant aunt in somewhere like India or Australia, but it won't tell you what diseases you're going to have. And that's what the study shows. So I say I'm sorry, because we've wasted 20 years when we could have been focusing at the level at which it's possible to understand health and disease. That's higher levels of the organism. And we didn't do that. We chose to spend all the money on sequencing as many genomes as possible. And we've ended up with the result that it doesn't tell us what makes us work. What makes us work as organisms is us. It is our cells, our tissues, our organs, and even the organism as a whole. Because remember, I put the sociotype at the top of that inverted triangle of causation. The really important decisions in life, like who are you going to marry? What kind of religion do you want to adopt? Do you want to adopt one at all? Or do you want to go on a holiday this way or that way? All these decisions are social decisions. They've got nothing to do with what genes you've got. Yes, there will be a propensity for people with particular kinds of genes to go one way or the other. But that's just probability. It doesn't tell you what will happen. So I think we can state categorically now that the idea that there is a program in the DNA and that those genes created this body and mind is simply illusion. It's fairy tale. And that's what the selfish gene is. It's a fairy tale. Actually, I told that to Richard Dawkins last year, and he replied saying, but Denis, if you're right, I and many others have been wrong for 50 years. I just nodded my head. Enough said.
Thank you, Dr. Denis Noble. I want to ask you why still the focus in biology is more on molecules, because we don't find the nature that you have suggested as organism possess, the religion, love, affection, all those things, properties, the subjective experiences, they are not coming from the physical properties or the chemical equations. There it is not represented, the happiness, unhappiness, love, or even knowing, even learning from the mistake what bacterium display. These qualities are not coming from those molecules. And why in biology we are still focused on molecules than finding a new way of representing life, or finding new terminology?
Well, I would respond to that by saying, first, let's give praise where it is due. The molecular revolution in biology, starting around 1950, well, exactly when you date it is arguable, but anyway, certainly coming to great fruition during the 1950s, when the double helix was first worked out, and when many other molecular processes were developed, such as how genes are read, how they're transcribed to create proteins, the one-way process of DNA to RNA to proteins, all of that. Let's give praise to the people who did that. They produced correct information, but it was misinterpreted for over 50 years, and particularly the idea of what is called the central dogma of molecular biology, which is the idea that because a DNA creates an RNA, which then is enabling a ribosome to make a particular protein, that there is a natural causation from the DNA through to the proteins, and therefore, because the proteins are the workhorses of the body, to what the body does. But I'll tell you this, if I exercise, as I do, I go and I spend exercising, doing walking, dancing, you name it, because I'm old enough to need to do that. You need to keep well. And exercise is one of the best ways of keeping well. What happens? If I maintain that, my muscles grow. How does that happen? That happens because that decision to exercise in that way leads to the exercise itself, which then sends messages to the right part of the genome to make more protein, actin and myosin, that enables muscles to move. All of that is correct. But does it tell you which way you will run? Does it tell you who you're going to meet? No, of course it doesn't. All it tells you is you will get more muscle made if you exercise, and that's all it will do. And so it's limited to what the central dogma actually states, which is that DNA leads to RNA, leads to proteins. And if you activate that process, you will get more of that particular protein. That's fine. But that certainly does not tell you where you will find the person you love, where you will find the person who you need, because you need a carpenter, for example, nor where you should go to have dinner with somebody. All of that is at a higher level of organization. So I think we should praise the founders of molecular biology for the brilliant discoveries they made. Without understanding the double helix, we wouldn't really understand how DNA gets replicated. Without understanding the processes of how RNAs are made and how that enables proteins to be made, we wouldn't be able to, to pursue possible ways in which we can cure diseases that depend on that process being much more functional. So, yes, praise where it is due, but the terrible misunderstandings that arose from taking the central dogma of biology to mean seriously what Richard Dawkins states, they, genes, made us body and mind. That is where the mistake is.
I want to emphasize something that is our Vedantic view is there is a gross body and there is subtle body. So these all protein that how it is synthesized, all this understanding more to do with the gross body, which in Vedic science is known as Panchamahabhuta: earth, water, fire, air, ether. This is basically the bodily constituent that we call about atoms, molecules, macromolecules, all those things. But what organism also displays the mind. In Vedantic sense, there are three more elements. They call it mind, intellect, and false ego. So at least there is some sense of mind. Some biologists have started to talk about as far as cell is concerned, as if cell does things as if it has some mind. The proofreading and stuff like that. So to address mind, only gross explanation or a study at the plane of gross level is not seems to be sufficient. So to include more subtle terminology which are being addressed in philosophy and religion is also taken into consideration if at all we want to address or justify the study of life.
I like that question very much indeed, because I think the mind obviously is in principle at that level I call the sociotype, and even bacteria have a sociotype, and certainly all single cell eukaryote organisms have a sociotype. They work out which organisms they can cooperate with and which they can't. So yes, it has to be at the level of the sociotype, and as I said, even single cells partake in a sociotype. Bacteria communicate to each other when they're hungry and when they're not, and they will let others know when they can reproduce and when it will be difficult. All of that is understood. I think on the question, though, where is the mind? I actually turn in my little book, The Music of Life, to the Oriental philosophical tradition for what I see as an easier explanation than Descartes' explanation given 300 or 400 years ago here in the West. Descartes thought we had to suppose there was a separate fuzzy thing, the soul, that communicates somehow or another with the brain. Well, we've never found that, and nor will we ever do so, because we come back now to what is happening at that high level. It's what I call conditioned arising, and some of you will recognize that already as Taoist, Vedantic, and Buddhist philosophy. It is the idea that at a high enough level of organization, all components of the system enter into determining what happens next. It is the process of conditioned arising. In Taoism, there's a very famous text from about 3,000 years ago that explains the idea. In Buddhism, of course, there's the no self concept, which I think is also found in other Indian traditions. I'm not a great expert on these Oriental traditions, but I do see them as having rich insights, which unfortunately here in the West we have ignored. And that's largely because we were impressed, I think, by Descartes' mechanism. He was living in a century, the 17th century, which was very impressed with the mechanical objects that looked like living organisms in the gardens of the rich. They even moved their hands like this. They even spoke, making mouthing noises and so on, all because of water being pumped through tubes that made the bodies move. And he thought everything has to be like that. He thought that fluid coming down a nerve would make a muscle expand and therefore contract, and so on and so forth. Of course, none of that is true. As soon as we found the electrical properties of nerves and muscles, we had no longer any need for Descartes' ideas. But his idea, and he wrote in 1665 in his treatise on the fetus, if I knew what was in that sperm, I would be able mathematically and with certainty to predict what would happen to the organism. There you have it. Even 350 years ago, the central dogma of molecular biology, which has been so badly misinterpreted as to think that that would be possible. It's just not possible. And as I said, the study by University College London in its recent investigation of the role of polygenic scores in predicting the human shows that Descartes was completely wrong. But we've had to wait 350 years before that test was done. It fails.
So now I'd like to thank this nice exchange and mention that Dr. Noble will be joining us at the end of the conference for the interdisciplinary dialogue. So any other questions for Dr. Noble or the dialogue amongst the speakers themselves as we continue to hear the ideas being presented today, we'll be able to have more of that later on. So we thank everyone and just like to transition now with Dr. Sumangala to our next speaker.